The task for the day, I think, was to, okay, how do you operate the, I really do know computers, just not this one. So I want to start off with acknowledgments because typically these get left to the end, and want to first thank Troy. I'm going to talk about some of the stuff that Troy did, and then at the end I'm going to sort of throw him under the bus also. And Troy told me to throw Bob under the bus, and I'll explain that later. I'm going to show, I decided rather than talk about some of the stuff that I personally have been working on to highlight some of the stuff that my students have been working on. And it all goes to the same picture I put up here. I deal with a lot of sequence data, and it's over a lot of different grants. So I don't just work in cows, although most people think I just work in cows. I work in a lot of different species. And I wanted to put this up here to sort of set the stage. And I realized this is the genetic prediction workshop. So this may be a bit of heresy. Honestly, I'm not really interested in genetic prediction. What I'm interested in is understanding how the genetic variation within individuals manifests itself as phenotypic variation. And you say, well, isn't that genetic prediction? No, they're two different things. So everything my lab has been focused on has been building tools and resources to get to that end goal that I'm interested in. I have a couple quotes here. Anybody that was at PAG 2008 when we rolled out the SNP 50 and Holstein started genetic prediction, Kurt said, we don't need no stinking genies. Anybody remember that? I barely do. But anyways, if you're just interested in genetic prediction, that's absolutely true. But I think in the ensuing 15 years, we've learned that not all variants are created equal. And I'll talk a little bit about that. Something else, I saw Mark, I don't know if you ever remember telling me this. This was back in the days of genotyping. And Mark said something very insightful over 20 years ago. He said genotypes are actually phenotypes. And what do I mean by that? Well, every one of us, every one of our animals has some inherent truth of the DNA sequence in that individual. And we use reproducible biochemical methods, whether it's SNP genotyping sequencing, to infer what the true underlying genotype is. And all of those things are measured with error. And then this last one, I like to always, it's one of my favorite quotes, I like to remind myself to let the data lead us to results, not our preconceived notions. And I may say some stuff that somebody might not agree with. Troy already covered this, and he did a really good job publishing this several years ago. And I tell people, you can trip over high imputation accuracy in the high minor loop frequencies. What I've been focused on in the last 4 years is how do we bend that curve? And that's what I'm going to talk about sort of today. Start off talking about sources of error. Troy mentioned a couple things. I'm going to talk about a couple things that you may not have thought about. How do we genotype SNPs and indels better? How do we actually genotype structural variation? And what is the landscape of this variation? And all of this builds to, well, we got to have these catalog of variants in order to do phasing. And then the imputation is actually the last step. Along Mark's comment that genotypes are actually phenotypes, and Troy already hit on this, the published genotyping accuracy for the Infinium assay is about 99.5%. And if you have 800,000 markers, that's 4,000 errors per individual. Most people don't realize that. It might be a little bit less, but there's approximately 4,000 errors per individual. If you look at sequencing for a Fred quality 30, it's 99.9% accuracy at a per base level. But if you have 20x coverage and 60 gigabytes of data, that's 60 million sequencing errors for every individual. The other thing I like to always remember or remind people is the infinium genotyping that everybody's using, that genotype's biallelic. So what happens if there's 3 or 4 alleles at a position? The answer is the genotypes are wrong. So I was interested in that, so I asked the question based on the 1,000 bull run 9 data, which were 6,192 individuals. If you look at that tranche level and say, how many multi-allelic positions are there in the genome? Because at the time, the 1,000 bulls run 9 was the best representation of cattle sequence data. It turns out that on the chips that we are genotyping, there's about 3% of the positions that have 3 or 4 alleles across all cattle that you look at. And that means that for those individuals that have an allele different from what was designed on the chip, the genotypes are absolutely wrong. So then the question becomes, well, does it really matter? Well, if you look at alleles that have a minor allele frequency greater than 1%, it probably doesn't matter. And if you are looking at genetic prediction, it probably really doesn't matter. But I'm not interested in genetic prediction. I'm interested in building reference panels and trying to get at the truth. This is something else that I talk about every time I get the opportunity. Cows are cows. And I haven't been able to definitively demonstrate it, but I will at some point because I'm getting closer to being able to do it. We need to stop thinking about breeds and think about cows. And this is something that the group at Roslyn published. Did that just get louder? Sorry. I'm getting different feedback depending on where I am. They made the point looking at pigs that the phenotypic variation is not due to unique alleles between different populations. It's due to differences in allele frequency of pre-existing variants. So I asked a very simple question. This was, I think, from 1,000 bull data. If you take the 4 or 5 largest number of breeds, pull those out, calculate the allele frequencies, and ask how many of them are unique to these breeds, the answer is hardly any. And as you sequence more individuals or sequence broader, you're going to find that the private alleles are the exception and not the rule. To Stuart's question, when do we throw away data? When do you reprocess it? I was really interested in sources of error, so I took the 1,000 bull data, and you got to remember from run 7, 8, and 9, when we switched over to the new reference, I asked the question, how many individuals were present in run 7 and run 8 and run 9? And the answer was 3,600. And if you say those data have not changed, the only thing that's changed is we've added more individuals and we perhaps tweaked how we process the data. I asked the question, if you consider the run 9 as the truth and compare for those 3,500 individuals, their genotype in run 9 to run 8 and then run 7 and then 8 to 7, how often do you get the same genotype? And this is looking at F1 precision and recall. And those numbers are scary low. To Stuart's question, do the genotypes change? Well, the DNA in the individual didn't change, but how we estimated their genotypes absolutely does change. So that led me to how can we do a better job at genotyping these things based on sequencing, and the GATK best practices workflow. The last step, variant quality score recalibration, is the hardest, and everybody just does defaults because it's hard, but I was not satisfied with that. So I did some work, a lot of work, to actually ask the question, can we better parameterize this and estimate this, come up with better truth sets, priors, and all that? And the answer was absolutely. What you see here in gray is the 1,000 bulls run 9 data. And what you see here in blue and orange are, the orange is the optimal. What you see is have lower Mendelian error rates up to a certain threshold, and then it changes. And it turns out that's the tranche level that you switch to pass variance, and it makes a huge difference. So the question then becomes, GATK best practices this is being recorded, isn't it? Yeah, GATK best practices variant quality score recalibration is a pain in the ass. So the question is, can we do better? About 4 years ago, Google released deep variant. And I had an informatics student, Jenna, at the time, was starting in informatics. I said, this looks really cool. I really like their model. I really like how they do it. Why don't you look into that? And that turned into a 3-year-long, really deep rabbit hole. But what deep variant does is it takes the image, or Google AI for image classification, and it transforms the genotyping into an image classification system. And it's based on how good are your truth sets. And in order to retrain deep variant, you got to have truth sets, and then you also have to have a method for redoing it. And to our knowledge, we're the first people to do it outside of humans. And the way this works is we start with the human deep variant checkpoint, and then we have a series of 14 trios, and we have GATK best practices call sets that we treat as truth, even though we know they're not. But they're our best estimate of the underlying genotype of that individual, and we train on dad, pick the best checkpoint by evaluating on the offspring. Once we have that best model checkpoint, then we select a new checkpoint. We have 19 individuals that are high coverage cows across various different breeds that we use for evaluation and compare the deep variant call genotypes versus our GATK truth set. And then we take that new checkpoint, start again, retrain on mom, evaluate on offspring, pick a new checkpoint, and you go around that loop through all 14 trios that we have. So the question is, does it work? Well, this slide is mainly to illustrate Jenna had different what we call phases for training. And it starts off here. So this is chronological as you start training. The T1 stands for Trio 1, Dad, evaluate, Trio 1, Mom. And in phase 1, we had 6 trios. I don't have time to discuss what constituted those, but you can see the different sort of phases. And I think there's various things. The color actually didn't show up when you see her, but basically the higher the value, the better. The less sort of bouncing around, the better. And you say, well, what are those big squiggly lines there? Those are the indels. Those are just always a pain in the butt. But what you can see is we sort of changed, and what I don't show here, this is starting at Trio 1. What I didn't show is what the deep variant F1 scores look like using the human model. This is a better representation of that. Similar, these are using the 19 individuals that the model has never seen to evaluate how good did the model do on data that it's never seen. And you start, there we go. You start at the top, it goes chronologically. These are the human models going down, and what's shown here is the F1 score, and the default out of the box human model that everybody is using based on our data as an F1 score of, I think that's 0.95. 0.95. Pretty good, but not that great. As soon as you train on the first cow trio, it goes up substantially. And if you squint really hard, you can see that these actually continue to increase as we are training. This is for indels. Indels are just a pain in the ass. Why are these worse than these? Well, because they're harder to genotype. Our underlying scare quote truth set for indels based on GATK is not that good. Pretty good. I would argue it's the best out there, but it's still not that good. So I was really impressed with that, and I said, Jenna, the problem is we don't have truth sets. All we have is our best guess at cow genotypes based on one genotyping model, GATK, and we're comparing a different genotyping model to that. Why don't you evaluate this using human data for which we actually do have truth sets? So she took her trio train pipeline, downloaded the human Han Chinese and Ashkenazi Jew trio, reprocessed the data through her pipeline, and what you see here is in these two human trios, using the human model, you have an F1 score 99.5. Really good. Using our cow trained model on human data, using the human genome in a bottle benchmarks, we have an F1 score of 99. That's pretty darn good. It's pretty amazing. We do much worse on indels simply because the model that we trained it on was trained on pretty shitty indel data. So that was really cool. But I said, Jenna wasn't happy with this and the F1 score, so she looked at Mendelian errors. And what you see here, these are three bovine trios that everybody's familiar with. This is the Angus Brahmin that was used for reference. This is the Yaplander, and this is the Bison Simmental trio. All of these have reference assemblies made out of them. We process the data, and the green here are the default human models that everybody's using, evaluated for Mendelian errors in our cattle trios. And what you can see is our cow model naturally does better in cow than what the human model does. The real surprise, though, was in this Jewish trio. We actually, our phase 4 model actually has fewer Mendelian errors than what the human trained deep trio model does. And we presented this a week ago Monday, and Andrew Carroll, if you know him, leads Google AI. He was actually really impressed with this. So we're going to be working with Google to further develop this. The bottom line is we need better truth sets. So if you need better resources, about the only way to do that is to write a USDA grant, convince somebody to give you money. And that's exactly what Jenna did. So she graduated last week. She's going to be staying on as a postdoc because she wrote this grant. She's going to lead this project. I don't have time to describe the Genome in a Bottle project. If you're familiar with it, we are doing that for cattle. And we have buy-in from the Human Genome in a Bottle group, and we are going to sort of brand this as Genome in a Bottle Ag here. We are going to focus on cattle. We have the resources, money, to do this in cattle, but all of the informatics that the human community has developed and all the informatics that we are going to develop as part of this, we are inviting all of the ag species. If you can generate this, we will do the rest for you. And the plan is within the next 3 years to have these genome in a bottle truth sets for all of the ag species. And I think that's going to revolutionize how we do things. So that's SNPs and indels. So using deep variant, we can do a really good job hauling SNPs. Kind of a crappy job doing indels, but better than otherwise. What about structural variants? Everybody, I think Troy mentioned it a little bit. I think Warren mentioned a little bit. there's a whole other class of variation that we're completely ignoring. And I'm going to break structural variants into copy number variants and then larger structural variants. My master's student who just graduated last year also, he did a copy number variant analysis based on SNP chip data. And these are the data that he had. These are the different assays. And there's several of you that are in the room that I call the Friends and Family Program who contributed genotypes to these. But in total, we had about 61,000 individuals with SNP chip genotypes across the various different assays and lots of breeds. Asked a very simple question: what does copy number variant landscape look like across cattle? Now, I'm not going to show you everything that he did because he doesn't have time. I'm actually going to get done. I'm going to get us back on schedule. Everything that's been done so far in copy number variant analysis as it relates to SNP chip data has been relatively small scale. 400 or 500 individuals would be a good study, maybe 1,000. When you start looking at 60,000, you get different pictures and you learn a lot. It's probably really hard to see this, isn't it? So I'm going to set this up and sort of tell the story that these slides show. This is chromosome 15 on the HD assay. X-axis is genome position along the chromosome, and then these are the various cattle breeds that we've included here. Every time you see a tick mark here, that's where a copy number variant or a CNVR was located. The height is represented by the frequency as well as the color. So it should be very apparent using this example that as you increase sample size, you detect more variants, right? Except for the frequent stuff. The frequent stuff that's in almost all cattle breeds, you're going to detect that with a small sample size. But it's not until you start getting larger and larger sample sizes representing more of the haplotypic diversity within each one of these animals do you start seeing all of these. What does it look like on the F250? Now, talk about preconceived notions. I went into this thinking that the HD assay with 800,000 markers would give the best representation of copy number variants across the genome, but I was wrong. I almost didn't let my student graduate because of it, because I thought it should be one way, and it turned out that it actually wasn't. And if you think about it, it has to do with the density and the marker spacing based on the algorithm that is using to detect this. And the F250 assay is not only functionally different, it is structurally different in terms of where we place those markers. It is highly, highly biased for exonic regions. And that allows you to actually detect smaller CNVs that are actually focused on individual exons. And what you see here, again, this is the F250, the same chromosome. As you have small sample sizes, you pick up the common stuff, and as you increase sample size, you see a lot more copy number variants across all of the genome. And it turns out that the F250, don't have the data on this because I just told him to do it last week. I am almost certain that we're going to identify copy number variants for individual exons across the genome because that's how we built the chip. But the overlap between what you detect on the HD, which has an average marker spacing of 2 kb, and what you detect on the F250 are completely different. So where I was wrong was assuming that we would detect the same things, and we don't, but it's just due to the way the assays were designed. So I had to make up a final, what we call a high confident copy number variant region list, about 1,300 that we are certain are real. They're present across a lot of individuals. They're at actually low frequency, but we're very confident in them. And the important number here is being very, very conservative of how we call these. We estimate at least 8% of the genome is involved in copy number variation. Why is that significant? Because it means that all of the SNP chip loci that we are using to genotype for individuals where those markers fall within copy number variation regions, our genotypes may not be right. Becomes really important when you start assuming that the genotypes are right and then using those to impute everything else. Another common theme is most of this variation is rare, much less than 5%. And that kind of surprised me. Now, you can clearly identify stuff that is more common, but across the board, it is rare. And there might be biological reasons for that. Purifying selection, sort of keeping these frequencies down. But I think there's actually a simpler explanation for it, which I'm not going to talk about right now. So that's copy number variation. You say, Bob, but SNP chip copy number variation, old technology, who really cares? We got sequencing. So Jenna's second part of her dissertation was to take the 7,000 genomes that we were working with and evaluate a new software program that came out earlier this year. It's called Q. It's out of the Broad Institute, and it does a similar deep learning type convolutional, I don't think it's a CNN, I forget exactly what it is, but it transforms the structural variant calling into an image classification system, and it was trained only on data, on synthetic data, 4 kb or larger. So we're talking about large structural variants. And I said, Jenna, this looks really cool. Based on the pipeline and the infrastructure you set up for TrioTrain to do deep variant, why don't you see if you can make this work? And within about a month and a half, she set up the pipeline and the process and actually had to go through and edit some of their code, update it to new Python. But she processed about 7,000 genomes in a little under 2 weeks. And this identifies, like I said, large 4 kb or larger variation. So what do the results look like? There's a whole lot on this page. I'm not going to talk about everything. Turns out that you have to do a lot of filtering to sort of weed out the chaff from what you believe. But it turns out for Q, they produce a estimated quality value. And based on the work that Jenna did, which I'm not showing here, their quality value that they estimate, it's a Fred scale, it actually is pretty accurate based on the data that we have, based on looking at Mendelian error rates. What's the bottom line? 6,000 or so genomes. If you look at the high coverage, defined here as more than 15x coverage, she identified 800,000 deletions, 172,000 inversions. I'm not going to read all these numbers. Heterozygous and homozygous for these classes. 800,000 heterozygous. So you say, oh, that sounds surprising. What's even cooler, though, are these 257,000 homozygous alternates. These are individuals that have chunks of their DNA, at least 4 kb, typically larger, where they don't have what is in the reference. Now, we have markers, SNP chip markers in those regions, right? And what happens when you impute SNP chip markers based on all current technologies? You get genotypes out. For these regions of the genome where these individuals don't have that DNA, you are absolutely making up data, and it's going to be wrong. Don't ask me how to fix it, right? I wasn't here to solve the world's problems. All I'm doing is pointing out issues that we absolutely know about, and the resources are going to be coming online to deal with that. average, these are the average number, 400 SVs or deletions per individual genome. You say, well, how accurate is that? It turns out that these numbers actually kind of mirrored what we were seeing with the SNP chip copy number variant analysis, because the SNP chip copy number variants are actually these deletions and duplications. It's just a different name for them. But she looked at it and plotted, here you have genome coverage, here you have the number of deletions per individual, and prior analyses tended to have a linear increase in the number of SVs discovered based on sequencing coverage. And you would think that, oh, Troy just got done telling you that more sequence data is almost always better. That is absolutely true. It is almost always better to allow you to genotype something more accurately, but you have to ask yourself, there's an underlying true number of structural variants in each one of these individuals. And as you sequence more data, you should converge at whatever that true number is. You shouldn't continue to identify new ones on a per individual basis. And what we're showing here is just based on the limited 6,000 or so genomes, we think we're topping off at about 400 to 500. What are these duplications or deletions per individual? We think that is biologically relevant, and we think that these data are actually turning out pretty good. So, estimation of ancestral allele. I'm going to throw a shout out to Hunter, who's in the back. She's a new PhD student with me. She's learning how to write code, and I said, well, in order to learn how to write code, why don't you figure out a way to estimate the ancestral allele if I give you a BCF file with thousands of individuals in it. And the idea here is we've sequenced, collectively we have sequence on these outgroup species. And if we genotype all of them together, then we've got estimates of allele frequency at every position in the genome. And the ancestral allele should be the most common allele in these outgroup species. So she's working on a program to actually do that at scale, and give you sort of some preliminary results. She's just working on chromosome 25, about 2 million calls that she made. And it turns out even being fairly confident in making these calls, over 80% of them turn out to be the reference allele. What does that mean? Well, it means that Dominette, the Hereford, is actually a really good representation of all bovids. And this is going to have downstream implications. And doing some simple cow math, once we do this on the whole genome, improve our sort of analysis, we estimate that we're going to be able to estimate the ancestral allele for about 140 million positions in the genome. You say, why does that matter for genetic prediction? Well, it doesn't, but if you're interested in identifying the underlying biological mechanisms of how a genotype translates into a phenotype, it becomes fairly intuitive how you can use the ancestral information for those. All right. I was charged with talking about building reference panels. Finally got to it. All of that was set up to get to this point. Phasing. Troy mentioned it a few times. I've been focused on phasing. Well, I showed you I focused on genotype being better genotypes, but those better genotypes went into phasing. The next step is phasing. And I firmly believe that if you do a better job phasing, doesn't matter how you do the imputation, you will do better. So I've spent the last 2 or 3 years of my life focused on how do we do this better. And I am using, I think, who was it, Stewart asked, well, what phasing algorithm did you use? I've just made an executive decision. I'm using ShapeIt 5. I like the ShapeIt model. I like the parameterization of it. I like the knobs that I can turn to adjust stuff. And another important thing is it scales sublinearly with the number of individuals, number of haplotypes. So if you give it more data on a per haplotype basis, it actually gets faster. Now, they've already shown this in the UK Biobank. I'm talking about 6,000, 7,000 genomes. UK Biobank, they did this with 500,000 genomes. But I said, ShapeIt has a lot of parameters and you can dial these things in and various different parameters you can choose. So before I launched into this, I tested this. I picked, I think, chromosome 25, and I'm evaluating the switch error rate based on changing all these parameters. And what you see is using the default parameters, you get a switch error rate of 0.155. Pretty darn good. Well, that falls about right here. So you can tune parameters and do a worse job. But what I'm showing here is you can actually do better than what the defaults are. Well, you say, well, the best model here is only 0.124. That's a difference of 0.03%. Not a big deal. Why is it worth the effort? Well, that 0.03% is actually 20% better than what you started with. And when you start talking about millions or hundreds of millions of genotypes and potentially hundreds of thousands of individuals, a 0.03% matters. So what did I do? Sequence phasing. I'm working with a little over 600,000 variants. I'm working with 212 million variants that passed my VQSR that I showed you before. So what parameters did I use for phasing? Two effective population sizes, 330,000. Be happy to debate those with somebody afterwards. Three different maps. A recombination map that was published by Lee Ma's group years ago. A LD map that I created myself. I forget how many markers it has on it. It's a ridiculous number. And then finally, a no map, which assumes a 1 centimorgan per megabase average recombination rate. And for each one of these parameter combinations, I did 5 replicates, right? Because this is an HMM-based model, and it starts off with a random seed. I changed the random seed, and the question is, if you do the exact same thing 5 times, do you get the same result? The answer is no. Sometimes you do better, sometimes you do worse. So it turns out every chromosome for these 212 million variants, I phased 60 times. Now, I don't have the results yet because this took about 3 or 4 million CPU hours, and it just got done about 2 weeks ago. But I can show you the picture. And I've showed this previously. If you start off and think about this, there is a true representation of individuals' chromosomes. And if you color code mom and dad like this, and then ask the question, how well do we phase the offspring? You should only have maybe 1 or 2 recombination events for those. You all sort of get that picture. So going back to, this is 1,000 bull run 8 data that was phased with Beagle. You can see a lot of switches. Turns out there's about 609 switches on that one. For Eagle, there was 405. So Eagle did better, but clearly not optimal. Notice the Mendelian errors. Those are actually, so it was introducing Mendelian errors. Or there were Mendelian errors present in the raw data. That's a problem. So these last two are based on the process that I just showed you. It's not even the optimal one. I just picked one of those different parameter sets because I knew it was going to be better. This is what it looks like. Exact same individual coming out of my phasing pipeline. Clearly, in that replicate, it was perfect phasing, maybe one switch over there. But visually, you can see it is drastically different. And if we can better phase 200 million variants, all of the chip-based stuff that we're going to be imputing to is going to be better. How much better? Well, so we also have SNP chip phasing that actually just got done last week. These are all of the assays that I'm working with. You can see the number of loci on each assay, and these are the number of samples with genotypes for each one of these assays that I'm working with. And I'm just going to point out the F250, the HD, and then the combination of the F250 and HD. These are probably, well, I know the F250 is the single largest collection of data anybody has because I built the assay. I know how many Illumina or GeneSeq manufactured, and I've got most of them. Same thing, 29 assays, changed the parameters. I varied the MCMC iteration length, varied the depth comment, how deep do you go searching for haplotypes. Again, 3 maps, 5 replicates each. It turns out for each one of these chips on each chromosome, it was phased 240 times. Don't have results. That data just got done 2 weeks ago, and this is where I'm going to throw Bob under the bus because Troy is going to be picking this up and building the imputation pipeline to do our 2-step imputation from these reference panels up to the phasing sequence reference panel that I just did. And Troy said he would have started working on it, but he had to plan some BIF meeting or something. I'm just kidding. So
he volunteered.
Yeah, he volunteered. So I'm not going to read all of these because I'm almost out of time. Sample size matters. More is almost always better. More across breeds is better than smaller numbers within breeds. And finally, cows are cows. These data are expensive. I think Troy has already illustrated. I think Warren's talked about it. I'm sure other people are going to talk about it. We have limited resources. We should be pooling all of this data and treating the genetic variation within these populations as all coming from the same source because it absolutely did. So with that, I will take any questions and sort of leave you with, I don't have results, but I'm fairly certain we are going to be able to bend that curve. That's all.
Well, how computationally difficult is some of this stuff going to be? I mean, are we going to be able to pipeline this?
So, so define who and what step of the process people are doing.
You know, I'm interested in I would be interested in commercializing this. How does a breed association get a sample from a member in processed and imputed and the data back in a timely fashion to make a selection decision?
So I can't speak to that. My guess is Jesse may talk to that at some point or somebody from Neogen. My focus has been on building the resources behind all of that, in which case to do what I just did is incredibly computationally challenging. But you only have to do it periodically. So Troy mentioned, well, how often do you re-impute stuff? Well, when better resources become available, how do you determine whether better resources are available? Well, you got to develop the benchmarks to be able to measure it. That's what I've been focused on. As associations or service providers.
So is it your position that this should be in the public domain?
It should what be in the public domain?
Well, you know, like some sort of a master phased, you know, reference population.
In a perfect world, yes. I have hundreds of thousands of dollars of compute invested from my own money to develop this. Before I make it publicly available, I have to do what I want to do with it, is a short answer. somebody gave me $500,000 to sort of redo this and perpetuate it, continue it, absolutely, I would make it public domain. I think I'm out of time. Thank you. Hey, it was real fun. I haven't been out in a long time, and it's really fun seeing a lot of faces, so we'll have to chat afterwards.